Today, I want to take you on a fascinating journey into the world of a well-known bacterial pathogen named Salmonella. Now, you've probably heard of Salmonella before. It's why you're not supposed to do things like eat raw chicken. And while this is definitely true, there's much more to this bacteria than simple food safety tips. So, Salmonella is not just one entity. It's actually made up of two major groups, or what we call serovores. One of these serovores is a common nuisance, but the other is a far deadlier threat. And this is where our work gets really interesting. So let's start with non-typhoidal salmonella. Non-typhoidal salmonella has really broad host range, meaning that it can infect many, many different species, ranging from humans to birds to lizards. It actually infects over 300 million people every year. That's like the entire population of the United States. And in humans, it causes what we call self-limiting gastroenteritis, or what's more commonly known as food poisoning. Unpleasant and uncomfortable, yes, but not usually super deadly. Now enter typhoidal salmonella. This one's a game changer. It actually evolved to only infect humans, and it causes a disease known as typhoid fever, which is so deadly that it actually kills up to 30% of the people that it infects if they don't get prompt treatment, and ends up killing about 200,000 people every year. So, unlike non typhoidal salmonella, typhoidal salmonella not only infects the human intestines, but can actually spread beyond the intestines into systemic organs, including the liver, gallbladder, and spleen. And here's the really unfortunate twist: it's rapidly becoming resistant to antibiotics. Now, picture this. Imagine a strain of deadly typhoidal salmonella that no drug can cure. It's unfortunately not science fiction. It's actually happening right now. In fact, scientists have recently found multiple extensively drug resistant, or XDR, strains of Salmonella typhi in Pakistan. And these isolates are spreading in that region of the world at a rapid rate. But this is why our research is so critical. We really need to be able to understand these bacteria at a molecular level to prevent another global pandemic from happening. Now, in my quest to unravel the secrets of Salmonella, I'm really interested by its evolutionary journey. So, how did this bacteria evolve from a relatively common foodborne nuisance to a potential killer? The answer lies in changes to its DNA. And so, by comparing the genomes of both non typhoidal and typhoidal Salmonella, we can begin to piece together a puzzle that spans its evolutionary trajectory. So, we're not just reading bits of genetic code on a computer. Instead, we're interpreting its history, its current strategies, as well as its future plans. So we're going to be looking at the evolution of Salmonella through two different perspectives. And here's where our work gets really interesting. So first, we're going to be using the latest advancements in sequencing technology to construct what are called random barcoded DNA libraries in different strains of Salmonella. We'll then be exposing all these different libraries to a variety of stressful conditions. So picture this. Once Salmonella actually gets into your body, usually through some kind of contaminated food, all of a sudden it's going to be challenged with many different stressful conditions, ranging from pools of acid in your stomach to a really strong and powerful human immune response, to sites of nutrient deprivation in your organs, to a ferocious onslaught of antibiotics during treatments. So, for Salmonella to actually cause successful infection, it needs to be able to survive and thrive in the face of these many stressful conditions. And so, how does Salmonella do this? This is the question that we're going to try to get at using our randomly barcoded DNA libraries. So we're planning to expose these libraries to dozens and dozens of these infection-related stress conditions. We'll then use a cutting-edge systems biology approach to rapidly identify weaknesses encoded in the genomes of typhoidal salmonella, which might make them more sensitive to stress. So overall, through this functional genomics approach, we're hoping to get really important clues for how to design novel therapeutics that can be used to combat these deadly infections. Second, we're also going to model these salmonella infections using human-derived intestinal organoids. So what is an organoid? You can essentially think of an organoid as a mini organ that was grown by taking a tissue sample from a healthy human donor, in our case, from their intestines. And importantly, these mini organs or organoids recapitulate many features of the human intestinal lining. As you might remember from before, typhoidal salmonella is human restricted. This means that it can only infect humans. Now, this also means that we cannot use any animal models of infection to study typhoid fever. So instead, we'll be using these human-derived intestinal organoids to model these infections. And we're really hoping that these experiments will give us an unprecedented view into how Salmonella interacts with human cells. It'll be like having a high-definition, slow-motion video of the infection process. So overall, through both our advanced sequencing techniques, as well as our innovative use of organoids, we're not just chasing a scientific curiosity. Instead, we're on a mission to eradicate these pathogens, to develop novel therapeutics that can outpace their rapid resistance to antibiotics, and to be able to accurately track and also predict the evolutionary trajectories of this diarrheal pathogen. This is truly a battle at the microscopic level, but it's a battle that we must win to safeguard our future from the threat of another global pandemic.